Okay, so my name's Luca, and this here is Shashivan, and today we're going to be talking to you about enzyme inhibition and its viability in the treatment of cancer. So as we all know, enzymes play crucial roles in cell survival. They catalyze various reactions throughout the cell that allow the cell to survive and to grow. What we're proposing to do today is inhibit one of these enzymatic processes so that we can reduce the cellular proliferation of cancer cells, whereas we still maintain healthy cell growth. So here is the 26S proteasome, which is the target of our inhibition. The 26S proteasome consists of two 19S caps and a 20S subunit. If we take a closer look at the 20S subunit, we see four stacked rings in a cylindrical shape. The two most outer rings are called the alpha subunits, and they serve a structural role, whereas the two inner rings are called beta subunits, and they serve a catalytic role. Current cancer treatments revolve around chemotherapy and radiation therapy, which although they are effective, they have a huge negative side effect as they are not specific to cancer cells, so they'll kill normal healthy cells in the body. We want to eliminate this by using bortezomib as an inhibitor of the proteasome to stop cancer cell proliferation while still allowing normal cells to function and grow. Shown here is a simplified diagram of proteasomal function in the cell. In orange, we have a protein that needs to be degraded, and it gets ubiquinolated, which targets it to the proteasome and allows for subsequent degradation. Now we're going to focus on the step of protein degradation. Shown here is the mechanism for protein proteolysis. It's a little difficult to see, but in green we have our protein substrate, in red we have water, and in black we have threonine 1 of the beta subunit. The first step involves nucleophilic attack of threonine 1 onto the protein substrate. Water mediates proton transfer in this process, but is not consumed. We form this intermediate called the tetrahedral intermediate. This tetrahedral intermediate has an oxidant that displays a negative charge. This oxygen will form a double bond with its adjacent carbon, which will result in ejection of the amino component of the substrate, or the N-terminal component. This new intermediate that forms is called an acyl enzyme due to the presence of an acyl group. This acyl group will then undergo hydrolysis, which will result in regeneration of threonine 1 so that it can be used in future reactions, as well as ejection of the acidic component of the substrate, or the C-terminal component. So now that we know how proteasomal cleavage occurs, we have to understand what the different forms of cleavage are. We have chymotrypsin-like cleavage, peptidoglutamyl peptide hydrolyzing cleavage, and trypsin-like cleavage. In chymotrypsin-like cleavage, we see cleavage occur after bulky hydrophobic residues such as isoleucine. In peptidoglutamyl peptide hydrolyzing cleavage, we see cleavage after acidic residues, and in trypsin-like cleavage, we see cleavage after basic residues. Today we're going to be focusing on the chymotrypsin-like and the trypsin-like cleavage, as these are necessary to understand the reaction kinetics of inhibition.
Alright, now the reaction kinetics of the proteasome protein degradation can be modeled using this equation, which is the Michaelis-Menten equation. Now what's important to know about this equation is in this reaction, the Vmax and Km of this equation are not kept constant. Instead, both Vmax and Km increase as a function of the protein substrate length. Now, this is the actual kinetics of inhibition. How the inhibition works is that in the beta subunit ring, there's two specific sites where the inhibitor goes. One is the chymotryptic site and the second is the tryptic site. The inhibitor goes and binds to the chymotryptic site and thereby inhibits its activity. But at the same time, this activates the tryptic site and slightly increases its level of activity. Now this can be represented graphically with this graph. Now on this graph, on the x-axis is the concentration of the inhibitor that was added, while on the y-axis is the relative velocity of the reaction. The relative velocity is found by taking the observed rate of reaction with the inhibitor and dividing it by the observed rate of reaction without the inhibitor. As you can see from this graph, where orange represents the chymotryptic rate of reaction and blue represents the tryptic rate of reaction, with an increasing concentration of the inhibitor, the chymotryptic rate of reaction is significantly decreased, while the tryptic rate of reaction slightly increases. The reason this leads to inhibition overall is because the chymotryptic reaction is the rate limiting step, and therefore abolishing that reaction inhibits the whole enzyme. Now that we know what the proteasome is and how it is inhibited, it's important to know how this can be used to treat cancer. Well, proteasome inhibition affects the cancer cells in a variety of ways. But we'll be focusing on three specific ones for the sake of time. Three we'll be focusing on is cell cycle arrest, apoptosis regulation, and then inhibition of angiogenesis. which are the three in the boxes. So I can see right now. All right. So starting with cell cycle arrays. The cell cycle is regulated by certain proteins called cyclins and CDKs. They can be seen in this diagram as the cell cycle is represented by the arrow, and the red box points out to some of the cyclin-CDK interactions. The cyclins and CDKs are interactions are extremely important and must be regulated in the cell, but cancer cells have a dysregulated cell cycle. What happens is that the levels of cyclin and CDKs need to be decreased or increased after each phase for the next phase to activate, and they are decreased through the use of the proteasome. Inhibition of the proteasome stops the degradation of cyclins and CDKs and therefore halts the cell cycle in one phase and doesn't let the cancer cell proliferate. The reason this impacts cancer cells and not normal cells is because normal cells have regulations of the cell cycle and therefore they can overcome this, while cancer cells have a dysregulated cell cycle and therefore are severely impacted by proteasome degradation. The second way that the mechanism is apoptotic regulation. So normal and cancer cells have two extremely important classes of proteins called pro-apoptotic factors and anti-apoptotic factors. In normal cells, these are kept at a good equilibrium, and when pro-apoptotic factors are raised, it can lead to cell death, while anti-apoptotic factors keep cell proliferation. Unfortunately, in cancer cells, to aid their survival, they suppress pro-apoptotic factors while keeping anti-apoptotic factors abundant, which leads to their cell proliferation. Through the use of proteasome inhibition, anti-apoptotic factors are decreased while pro-apoptotic factors increase in the cancer cells, leading to cell death. The way it works, to explain that, we'll use a specific example. One extremely important pro-apoptotic factor in cancer cells is the p53 molecule. Now, cancer cells have a unique mechanism where they overexpress the MDM2 inhibitor, which is the inhibitor of p53, and that inhibitor binds to p53 and targets a degradation from the proteasome, and therefore the cancer cells are continuously kept alive. But once the proteasome is inhibited, the levels of p53 cannot be decreased, and it accumulates in the cell, leading to cell death. The reason this doesn't affect normal cells is because p53 is called the tumor suppressor molecule, and it only causes apoptosis of cells which have DNA damage, such as cancer cells. Increasing this level will not affect normal cells under normal circumstances. Now, the third and final way we're going to talk about proteasome inhibition helps kill cancer cells is through inhibiting angiogenesis. Angiogenesis is the creation of new blood vessels from pre-existing blood vessels, and tumors release angiogenic factors such as VEGF to continuously increase the amount of capillary formation to them, so they can get more nutrients from the body and increase the pathways for metastasis. But proteasome inhibition stops the release of angiogenic factors such as VEGF. The exact mechanism for how this occurs is not known. And now, to summarize our presentation, traditional methods for cancer treatments can be improved as they have multiple side effects. Bortezomib, which is a proteasome inhibitor, can be used to inhibit the 26S proteasome and thereby treat cancer. And inhibition of the proteasome can inhibit cancer through cell cycle arrest, apoptosis regulation, and angiogenesis inhibition, and many other pathways. And that's our presentation.
Yeah, perfect timing. 8.5 minutes. Great.